Hey you guys, I'm going to read topic 3, underline some important information, and make some annotations on the side of the text as I read. Topic overview. When two organisms reproduce, their offspring receive genetic instructions called genes. From each parent. The genes determine which traits or characteristics each offspring will have. All organisms, whether they are animals, plants, or members of one of the other kingdoms, pass their genetic characteristics along in this manner. Because of this transfer of genetic information, offspring tend to resemble their parents. Heredity and genes. Heredity is the passing of genetic information from one generation to the next through reproduction. The heredity information, DNA, is organized in the form of genes located in the chromosomes of each cell. The hereditary information is organized in the form of genes located in the chromosome of each cell. Recall that chromosomes, which are formed by the cell nuclei, contain the DNA molecules. It is in the DNA that carry the genetic information of the cell. A human cell contains many thousands of genes in its nucleus, and each gene carries a separate piece of coded information. The traits inherited by an individual can be determined by one pair of genes or by several pairs of genes. It is also true that a single gene pair can sometimes influence more than one trait. Table 3.1 shows several examples of these variations. Some traits that an organism inherits are readily observable. These include traits such as hair color, leaf shape, flower scent, and wing structure. The overall structure of the body is also an observable trait that is inherited from the parents. Some children, for example, inherit long, slender toes or large earlobes. Other traits are not so obvious. Less obvious traits may involve a defective heart, a single kidney, or how some of the body's chemicals function. Examples include the ability to produce insulin, the types of receptors present on a cell membrane, and whether an individual can make a particular respiratory enzyme. Methods of reproduction. There are two common methods of reproduction, asexual and sexual. The major difference between these two methods is whether one or two parents are involved in producing the offspring. Asexual reproduction involves one parent or individual. Sexual reproduction involves two parents. Asexual reproduction in organisms that reproduce asexually, all the genetic instruction genes come from one individual or parent. Since the genes are all from one parent, offspring are usually identical to the parent. Because the coded instructions in their cells are the same as the instructions in their parent's cell, asexually produced offspring are genetically identical to their parent. Identical genetic copies are known as clones. Because they are sexually produced, entire populations of bacteria, perhaps millions of cells, may be genetically identical clones. Sexual reproduction. In organisms that reproduce sexually, two parents are required to produce offspring. Each parent produces sex cells. Sperm are the sex cells produced by the male. The egg is the sex cell produced by the female. So I'm going to note that there. Male equals sperm. Female equals egg. Recall that genes in body cells. Cells other than sex cells occur in pairs, but each sex cell contains only one gene from each pair. The offspring that results from sexual reproduction therefore receives half of its genetic information from the female parent via the egg, and half from the male parent via the sperm. Genetic recombination. When a sperm and egg combine to form a new cell with a complete set of genetic instructions, a unique combination of genes result. The term for this is genetic recombination. This unique combination of thousands of genes produces an offspring that may resemble either or both parents in many ways, but will not be identical to either of them. Number 1. A cell is represented on the diagram below. Which statement about the cell is correct? 1. Structure A synthesizes and secretes cellular products. 2. Structure B contains chromosomes involved in transmitting genetic information. 3. Structure C utilizes DNA in the process of photosynthesis. Or 4. Structure D is the site of protein synthesis. 2. Which materials are composed of DNA? 1. Proteins, 2. Genes, 3. Nerve secretions, or 4. Fluid in vacuoles. Number 3, in an animal cell, DNA is found in the greatest concentration in the vacuole, ribosome, nucleus, or cytoplasm. 4. Cystic fibrosis is a genetic disease. Examine the illustration below. The mother cells most likely contained 1. A disease-causing virus, 2. One normal gene and one abnormal gene, 3. Two normal genes, or 4. An abnormal number of chromosomes. 5. Bacteria in culture A produce slime capsules around their cell walls. Biologists remove the DNA from some of the bacteria in culture A. He then injected it into bacteria in culture B, which normally would not produce slime capsules. After the injection, bacteria with slime capsules began to appear in culture B. What conclusion could best be drawn from this investigation? 1. The bacteria in culture A are mutations. Bacteria reproduce faster than they have slime capsules. 3. The slime capsules of bacteria in culture B contain DNA. 4. DNA is most likely involved in the reproduction of slime capsules. Number 6. The letters in the diagram below represent genes on a particular chromosome. Gene B contains the code for enzymes that cannot be synthesized unless gene A is also active. Which statement best explains why this can occur? 1. A hereditary trait can be determined by more than one gene. 2. Genes are made up of double-stranded segments of DNA. 3. All the genes on the chromosome act to produce a single trait. Or 4. The first gene on each chromosome controls all the other genes on the chromosome. Number 7. Which cell structure includes all of the others? Nucleus, gene, DNA, chromosome. The genetic code. The inherited instructions genes that are passed from parent to offspring exist in the form of a chemical code. This genetic code, as the chemical code is called, is contained in the DNA molecules of all organisms. DNA molecules resemble a flexible twisted ladder formed from many smaller repeating units. DNA structure. Like other large molecules of life, the DNA molecule is made of thousands of smaller sections called subunits. Each subunit has 3 chemical parts: a sugar, a phosphate, and a base. The subunits vary from one another according to the kind of bases they contain. The bases are represented by the letters A, G, C, and T. The 4 subunits of the DNA molecules are arranged in pairs, each subunit forming one side and half of one rung of the twisted ladder. Base A of one subunit always pairs with the base T of another subunit. In a similar way, base G always pairs with base C. Figure 3.3 shows the details of the structure in an untwisted molecule. Once the chemical and structure proteins of DNA were discovered by scientists, it became clear how this molecule could contain a kind of message that functions as a code. Notice in figure 3.3 the sequence of the bases on the molecule's left strand, reading from top to bottom, is ACAG. A different molecule might have a sequence in the same position reading GCAG or AACG. The specific sequence of bases in a DNA molecule forms a coded message. The message of a single gene is often a sequence of hundreds of bases. The code for an entire human is estimated to be about 3 billion base pairs. DNA replication. The ability to copy the coded instructions in the DNA molecule is critical to its function. Knowing the chemical makeup and structure of DNA molecules gave scientists an immediate clue to how the molecule could be copied or replicated. When scientists realized that the bases used weak chemical bonds to pair with each other, they also realized that the DNA could separate at that weak bond to form 2 single strands. Each single strand became a template or pattern for a new molecule. The new molecule was built by attaching new subunits to each template strand, always following the base pairing rule of linking A with T and C with G. So I put that over there. A goes with T, C goes with G. The result is the formation of 2 new molecules whose base pair sequences are exactly alike. When the structure of a DNA was determined, scientists finally understood how cells could copy and transfer information to new cells each time they divide and to new offspring during reproduction. Replication produces 2 identical copies of a cell's genetic information, each ready to be passed from the cell parent to the offspring cells during cell division. Offspring cells are commonly called daughter cells. Proteins and cell functioning. The work of the cell is carried out by many types of molecules the cell assembles or synthesizes. Many of these molecules are proteins. Protein molecules are long chains. They are formed from various combinations of 20 kinds of amino acids, arranged in a specific sequence. The sequence of amino acids in a particular protein influences the shape of the molecule. This is because some of the amino acid parts are attracted to and may bond with other amino acid parts of the chain. The connections that form between two parts of the chain cause it to fold and bend in a specific way. The final folded shape of the protein enables it to carry out its function in the cell. Many proteins made by a cell become enzymes that regulate chemical reactions. Remember that an enzyme can interact with a specific molecule because their shapes correspond. Some of the proteins made in cells become parts of organelles, such as the cell membrane. Other proteins include the hormone insulin or many antibodies that bind to antigen molecules on pathogens. The color of your eyes and skin are also the result of proteins synthesized by your body. The DNA-protein connection. Cells store vast amount of coded information in their genes. Much of this coded information is used to make the thousands of proteins that each cell requires for its functions and the structures it contains. The proteins for these structures and functions are made at the ribosomes according to the directions stored in the cell's DNA code. So that's going to be important. Ribosomes make protein. Because offspring inherit genetic information from their parents, their cells make many of the same proteins. This is what causes the resemblance between some children and their parents. Making many of the same proteins causes both parents and offspring to form similar structures that give them similar features. One example of a protein-dependent trait includes her hair texture, curly, straight, or kinky. If a parent's DNA carries a code for a protein that does not function correctly, the children may also make that defective protein. For example, an albino does not produce the usual amount of eye hair or skin color pigment. The condition is caused by a defect in the gene that codes for the protein that produces color pigment. If albino parents pass the gene to their offspring, they too may not produce the normal color pigment. Protein synthesis. The process of synthesizing a protein from DNA begins in the nucleus. There, the DNA code of a particular gene is read by a special enzyme and used to produce a messenger molecule. This messenger molecule then travels to ribosomes in the cell cytoplasm. With the aid of specialized transfer molecules, amino acids are moved to the ribosomes for assembly into protein. They are bonded in the order specified by the messenger molecule. In this way, the sequence of amino acids of any protein, and therefore its overall structure, is determined by the gene's DNA sequence in the nucleus. The process is shown in Figure 3.5. Review questions. Number 8. In a DNA molecule, the letters A, T, C, and G represent bases, sugars, starches, or proteins. 9. The individuality of an organism is determined by the organism's amino acids, nitrogen bases, DNA base sequence, or order of ribosomes. 10. What would most likely happen if the ribosomes in a cell were not functioning? 1. The cell would undergo uncontrolled mitotic cell division. 2. The synthesis of enzymes would stop. 3. The cell would produce antibodies. Or 4. The rate of glucose transport in the cytoplasm would increase. 11. The diagram below represents a portion of a DNA molecule. The letter X represents two bases that are identical and joined by weak bonds, identical and joined by strong bonds, a part of the genetic code of the organism, or amino acids used to build folded protein molecules. Number 11. The kinds of genes an organism possesses are dependent on the type of proteins, the organism's nuclei, sequence of bases in the organism's DNA, number of ribosomes in the organism's cytoplasm, or size of the mitochondria in the organism cells. Number 13. What is the role of DNA molecules in the synthesis of proteins? 1. They catalyze the formation of bonds between amino acid. 2. They determine the sequence of amino acids in a protein. 3. They transfer amino acids from the cytoplasm to the nucleus. Or 4. They supply energy for protein synthesis. 14. The diagram at the right represents a molecule of 1 ATP, 2 protein, 3 carbohydrate, or 4 DNA. Number 15. During replication, the strands of a double-stranded DNA molecule separate when the bonds are broken between their paired bases. Explain why, in terms of the genetic code, it is important that the molecules separate between the bases and are not at some other point. Base your answers to questions 16 and 17 on the diagram below and on your knowledge of biology. 16. Within which organelle does process A occur? Ribosome, nucleus, vacuole, or cell membrane? 17. Within a living cell, which organelles are directly necessary for process B to occur? Ribosomes, mitochondria, vacuoles, or cell membranes? 18. In all living things, DNA controls cellular activities by 1. Determining the order of amino acids and protein molecules, 2. Regulating the concentration of molecules on both sides of the cell membrane, 3. Varying the rates of starch synthesis, or 4. Coordinating active and passive transport. 19. Which cell organelle indicated in the diagram control the synthesis of enzymes? A, B, C, or D? Number 20. The sequence of amino acids that makes up a protein molecule is determined by the sequence of bases in DNA, glucose in DNA, ribosomes in the cytoplasm, chloroplasts in the vacuole. 21. In DNA, the base represented by an A always pairs with the base represented by A, T, C, or G. Number 22. The presence of DNA is important for cellular metabolic activities because DNA directs the reproduction or production of enzymes, is a structural component of cell membranes, directly increases the solubility of nutrients, is a major component of the cytoplasm. Mutations. Genes are actually segments of DNA molecules. Any alteration of the DNA sequence is a mutation, which changes the normal message carried by the gene. Many mutations involve the substitution of one base for another. This often causes a different amino acid to be placed in a particular position in the growing protein chain. Some mutations involve the insertion of an additional base into an existing DNA sequence. This affects all of the code past the change, just as skipping a blank on an answer sheet for a test can cause all the remaining answers to be shifted to the next blank, making almost all of them wrong. The deletion of a base from the normal gene sequence would also alter all the code past the gene. Some mutations occur when the bases within a gene are accidentally rearranged. This too alters the genetic code. Figure 3.6 shows several ways that DNA can mutate. All of these alterations are totally random and can occur anywhere along the molecule, making the result of the DNA almost impossible to predict. However, when a DNA sequence is changed, it is quite likely that the protein it codes for may be assembled incorrectly. If some amino acids are replaced by others, or if their sequence is different, the folding of the protein may be different. Incorrect folding means that the protein shape would not be normal. This could cause the protein to malfunction. One mutation caused by substitution is sickle cell disease. Mutations can cause serious changes to the cell that may die. However, a mutated cell does survive and can replicate its DNA. Its changed instructions will be copied and passed on to every cell that develops from it. In sexually reproducing organisms, only mutations found in sex cells can be inherited by the offspring. DNA and individuality. Although an individual's body cells all originally come from a single cell, the body is made up of many types of cells. Each body's cell nucleus, whether it be a nerve cell or a skin cell or a bone cell, has a complete set of identical genetic instructions for that individual. For years, scientists wondered how cells with identical genetic instructions could be so different. The answer is that each kind of cell uses only some of the genetic information it contains. It uses only the instructions it needs to operate its own kind of cell. For instance, information for building all of a person's enzymes is coded in the chromosomes of each cell. But a muscle cell uses only the specific enzymes that are needed by a muscle cell. Both of the internal and external environment of a cell can influence which genes are activated in the cell. Some of this influence may occur during development, leading to the many different types of cells that an organism needs. The selective activation of genes in a cell may continue as conditions change throughout life. For instance, chemical signals from within the cell or from other cells may activate a particular gene. Hormones are one kind of molecule that can activate parts of a cell's DNA code, leading to the production of a particular protein. Although genes are inherited, an organism's environment can affect the way some genes are revealed or expressed. In the organism, for example, in some animals such as the Himalayan rabbit, the outside temperature can cause the activation or inactivation of the genes for fur color. When the rabbit's body area is cold, black fur grows. If the same body area becomes warm, white fur grows instead. The environment can also influence human genes. Studies of identical twins, those with identical genetic information, who are raised in different environments, show that they have differences that can only be explained by the influence of the environment or gene expression. Review questions 23. A dog breeder can determine the sudden appearance of a hairlessness in one of the puppies as a mutation of the dog. 1 is still hairless after 5 years. 2 shows no change in the hairless condition after its diet is changed. 3 develops other conspicuous differences from the parent. Or 4 is bred and the trait is capable of being inherited. Number 24. Explain how a change in the sequence of nitrogen bases in a DNA molecule could result in a gene mutation. 25. Which mutation could be passed on to future generations? 1. A gene change in a liver cell. 2. A cancer caused by excessive exposure to the sun. 3. A chromosomal alteration during gamete formation. Or 4. Random breakage of chromosome in a leaf cell of a maple tree. 26. Mutations can be transmitted to the next generation if they are present in 1. Brain cells, 2. Sex cells, 3. Body cells, or 4. Muscle cells. 27. Overexposure of animals to X-rays is dangerous because X-rays are known to damage DNA. A direct result of this damage in cells with unusually thick cell walls, no organelles located in the cytoplasm, abnormally large chloroplasts, or changes in chromosomal structure. The diagram below shows a portion of a DNA molecule. The base sequence of the unlabeled strand shown in the diagram is most likely 1. GAGT, CUCA, TCTG, or GAGU. 29. The individuality of an organism is determined by the sequence of bases in DNA, number of amino acids in a cell, position of ribosomes in the cytoplasm, or number of bases in the mitochondria. Number 30. In which situation could a mutation be passed on to the offspring of one of the organisms listed in the data table below? 1. Ultraviolet radiation causes fruit fly wing cells to undergo uncontrolled cell division, resulting in cells with 9 chromosomes. 2. A cell in the wall of the human uterus undergoes a change resulting in cells with 47 chromosomes. 3. A primary sex cell in a human forms a sperm that contains 23 chromosomes. Or 4. A cell in the ovary of the fruit fly undergoes a chromosomal change that results in 5 chromosomes per egg cell. 31. A change in the sequence of bases in a DNA molecule is most accurately referred to as 1. An insertion, deletion, or substitution. 2. A chromosomal replication. 3. Carbohydrate molecule synthesis. Or 4. Selective breeding. Genetic engineering. Genetic engineering is a new technology that humans use to alter the genetic instructions in organisms. The idea of altering organisms to have more desirable traits, however, is not new. In fact, biotechnology, the application of technology to biological science, has been producing useful products for thousands of years. Cheese and bread are 2 examples of biotech products made with the use of microbes. Throughout recorded history, humans have also used selective breeding, a process that produces animals, domestic animals, and new varieties of plants with traits that are particularly desirable. Many meat products, for example, come from animals that have been bred to contain less fat. In addition, many of the fruits and vegetables we consume have been selectively bred to be larger, sweeter, hardier, or even juicier. To breed a better plant, farmers might select a bean plant that produces many pods and then crossbreed it with a bean plant that resists fungus infections. The farmers would expect to get seeds that would grow into bean plants with both features. Gene manipulation. In recent years, plants and animals have been genetically engineered by manipulating their DNA instructions. The result of this genetic manipulation is new characteristics and new varieties of organisms. Consequently, we have been able to produce plants with many beneficial traits. In one instance, plants can now contain genes with instructions for making chemicals that kill the insects that feed on them. Scientists have also engineered bacteria that can be used to clean up oil spills or that produce human growth hormone. The basic method that alters genes in organisms uses special enzymes. These enzymes cut DNA segments in a way that allows the segments to be spliced or moved and attached to the DNA of new organism. Once in the new organism, the transferred genes direct the new organism's cells to make the same protein product as the original organism. For example, when we move a human insulin-producing gene into a bacterial cell, the bacterium and all its offspring will produce human insulin. This provides a way to produce large quantities of a hormone at a low cost. Genes for other human proteins have also been inserted into bacterial cells, as illustrated in Figure 3.9. Other enzymes have been found that can be used to make many copies of segments of DNA. These can be used to increase the amount of DNA available from a tiny sample. This procedure is helpful even when only a drop of blood or saliva is found at a crime scene. By copying and recopying the DNA in the sample, criminal investigators can produce a sample that is large enough to test. The test results may identify or clear suspects. Applications of biotechnology. The healthcare field has much to gain through our increasing knowledge of genetics and biotechnology. New methods enable us to locate and decode genes that cause diseases. Once we have been better understanding of the gene-specific defect, we can be able to develop ways to treat the disease. In some cases, we may be able to alter the DNA in affected cells and cure the person. Due to mutations in their genes, people with genetic diseases are sometimes unable to produce certain hormones, enzymes, or other body chemicals. At times, we can extract these chemicals from animals such as sheep and cattle. These extractions, however, can be expensive, and the chemicals may contain contaminants that cause side effects. If scientists can produce the chemicals used genetically engineered organisms, we may be able to economically provide the missing chemicals in a pure enough form to avoid the side effects associated with chemicals obtained from animal sources. Number 32. Genetic engineering is used in the biotechnology industry to eliminate all infections in livestock, synthesize hormones such as insulin and human growth hormone, increase the frequency of fertilization, or eliminate asexual reproduction. 33. Describe 2 examples of how an understanding of genetics is making new fields of healthcare possible. 34. The insertion of a human DNA fragment into a bacterial cell might make it possible to 1. For the bacterial cell to produce a human protein, to clone the human that donated the DNA fragment, for humans to become immune at an infection by this type of bacteria, or to clone this type of bacteria. 35. In a DNA sample, 15% of the bases are thymine. What percentage of the bases in the sample are cytosine? 15%, 30%, 35%, or 85%? Base your answers to questions 36 through 40 on the passage below in your knowledge of biology. Advances with cells and genes. Recent advances in cell technology and gene transplanting have allowed scientists to perform some interesting experiments, including splicing human DNA into the chromosomes of bacteria. The altered bacteria express the added genes. Bacteria reproduce rapidly under certain conditions. This means that bacteria with the gene for human insulin could multiply rapidly, resulting in a huge bacterial population capable of producing large quantities of human insulin. The traditional source of insulin has been the pancreases of slaughtered animals. Continued use of this insulin can trigger allergic reactions in some humans. The new bacteria produced insulin is actually human insulin, As a result, it does not produce many side effects. The bacteria used for these experiments are E. coli, which are found in the digestive system of humans and many other animals. Some scientists question these experiments and are concerned that altered E. coli may accidentally get into water supplies. For each of the statements below, write the number 1 if the statement is true, according to the passage, the number 2 if the statement is false, according to the passage, or the number 3 if not enough information is given in the passage. 36. Transplanting genetic material into bacteria is a simple task. 1 for true, 2 for false, or 3 for not enough info. 37. Under certain conditions, bacteria reproduce at a rapid rate. 1 for true, 2 for false, 3 for not enough info. 38. The continued use of insulin from animals may cause harmful side effects in some people. 1 for true, 2 for false, 3 for not enough info. 39. The bacteria used in these experiments are normally found only in the nerve tissue of the humans. 1 for true, 2 for false, 3 for not enough info. 40. Bacteria other than E. coli are unable to produce insulin. 1 for true, 2 for false, 3 for not enough info. 41. The headline, "Improved Soybeans Produce Healthier Vegetable Oils," accompanies an article describing how a biotechnology company controls the types of fats present in soybeans. The improved soybeans are most likely being developed by the process of natural selection, asexual reproduction, genetic engineering, or habitat modification. A product of genetic engineering technology is represented below. Which substance was needed to join the insulin gene to the bacterial DNA as shown? 1, a specific carbohydrate, 2, a specific enzyme, 3, hormones, or 4, antibodies. Number 43, last question. In the past, diabetics used horse or cow insulin to control their glucose levels. Today, as a result of genetic engineering, human insulin can be synthesized by bacteria. Say one advantage for a person with diabetes to receive genetically engineered insulin rather than insulin taken from a horse or cow.